This is Dr. Martin's supplemental lecture on Chapter 18. I'm going to start with Section 2. We were talking last time about fungi, so I introduced the fungi with some general features or characteristics. And now we're going to get into the separate groups or individual groups of fungi. So we have 4 groups that I'm going to talk about. There are more, but we're going to focus on the most important. One group are informally called the zygote fungi, at least from your textbook. These are technically called the Zygomycota. And this includes the common bread mold. A second group is informally called the sac fungi. And the technical term for this group is Ascomycota. A third group is the group that we looked at in lab on Tuesday, and these are called the club fungi. And their technical name is Basidiomycota. And the very last group are informally called the yeasts, and many of these are technically part of a group called the imperfect fungi. So we're going to focus primarily on these 3. I'll talk a little bit about the last group, but we'll start with the zygote fungi. And I'm going to use a figure from your textbook, and this is figure 18-4. All right, so these are, as I mentioned, the zygote fungi. Some characteristics or features here before we get into part of the life cycle. This is a group that includes the common bread mold. This is a primarily terrestrial group found in moist soils as well as decaying plant and animal matter. And it has both a sexual and an asexual reproductive cycle. Okay, a characteristic here of the 2 different types of reproduction. What happens is that when there is abundant food available, the fungus will actually go through an asexual phase, asexual reproduction. When there is a lack of food, or food suddenly becomes scarce, the fungi will go through an asexual reproductive cycle. So let's take a look at that. And we're going to start with the asexual, which actually is not shown here. This is, as you can see, the sexual reproductive cycle. So the asexual cycle again occurs when food is abundant. The fungi will produce spores on the top of stalks, and the spores are located in structures that are called sporangia. This is the stalk. This is the rest of the mycelium. Spores will be released by wind or rain from the sporangium, and those spores can germinate. So this is germination to produce more mycelium. I should also point out that this phase is what we call a haploid phase. And if you remember from genetics, this means that there's only one copy of each chromosome located in these cells. Okay, the sexual reproductive cycle begins when food becomes scarce. And what will happen is, in this case, we actually have to have mycelium from two different, we call them mating types, and they will come together. So here you see part of a mycelium from one mating type, and it's indicated with the red nuclei. This is a mycelium from a second mating type, shown here with black nuclei. The mycelium will start to grow toward one another, and they form special structures. Here we have those structures that are going to actually join here in the very center. So this is where we see fusion, and we produce a structure in the center that's called a zygosporangium. It has a hardened outer wall. It can withstand harsh conditions for some time. When conditions improve, this structure will sprout a stalk, and at the top of a stalk is a structure that contains lots of spores, and those spores can be released to generate mycelium. Now the point here is that at this point this is a diploid structure, so the cells now have both nuclei from both mating types in the same cell. When conditions improve, that will stimulate meiosis. And remember, during meiosis, we get segregation of the chromosomes. So now we're down to haploid spores, which again will, if they land in a suitable environment, those will germinate. And we can go back through the asexual cycle again. Okay, that's the end of part 1, and I'll continue with part 2 in the second lecture.